My name is John Mascarenhas. I'm a professor of medicine at the Icahn School of Medicine at Mount Sinai in New York City. So we're going to talk about the SENTRY study. This is a randomized phase 3 global study of selinexor plus ruxolitinib versus placebo plus ruxolitinib in JAK inhibitor naive myelofibrosis patients with a platelet count of at least 100,000, DIPS intermediate 1 to high risk of disease, and measurable spleen and symptom burden. The patients were randomized in a 2-to-1 fashion to the combination, which is rational and has preclinical and phase 1 data for it, versus single agent Rux plus placebo. The primary endpoint of SVR35 was measured at week 24, and the mean absolute change in TSS from week 0 to week 24 is the second co-primary endpoint. So what we're going to review at the oral abstract session is the initial results of the study. So this was a positive study from an SVR standpoint. Approximately 50% of patients in the combination therapy achieved an SVR 35% or greater, versus about 28% in single agent ruxolitinib. This is aligned with what we've seen in other trials, so it's quite consistent. The total symptom score improvement was the same between both arms. It wasn't statistically significant, but different. But there was an improvement that was appreciable and meaningful from baseline to week 24 with the combination. So although it didn't meet statistical significantly difference between the two arms, it was comparable as single agent ruxolitinib. And this is what we've seen with multiple early phase or phase 3 studies in the treatment naive setting. So it's quite consistent. There are other endpoints that are quite interesting. And I should point out that the SVR35 kinetics or the spleen volume reduction kinetics, I should say, with the combination were distinct. They were more rapid, deeper, and durable with the combination than with single agent ruxolitinib. And what we saw, based on the fact that the responses did seem deeper and more sustained with the combination, was a survival signal favoring the combination with a hazard ratio of 0.43. Now, this is early on without many events yet, so it needs to be followed through, but very encouraging. Interestingly, intriguingly, and probably very important to the field, this survival advantage was associated with an SVR35 at 24 weeks. So associating this early outcome measure of spleen response with an improved outcome of survival later on. Again, we'll have to continue to follow the patients, see if the survival is maintained, but that's quite compelling. Additionally, another biomarker favored the combination. That was reduction in driver VAF at 24 weeks, which occurred in about 32 to 34% of patients with the combination versus about 24% with single agent ruxolitinib. So this difference was not just in spleen response and the kinetics of spleen response, but it was also in terms of biomarkers that are surrogates for disease burden with a higher degree of reduction in driver VAF with the combination. And that did associate also with spleen volume. So it really all ties in together, linking reductions in driver VAF, improvements in spleen that are both rapid, deep, and durable, and the survival benefit that will need to be followed out more with longer-term follow-up. Let me add one more thing to the SENTRY study, which I should point out. We've talked about the efficacy in terms of spleen response and symptom improvement, survival benefit early on. That's quite intriguing and exciting. Reductions in driver VAP. I should talk about safety. So there is more GI toxicity and hematologic toxicity with the combination, as one would expect based on the profiles of the two drugs, and the phase 1 data in 18 patients previously would tell us that would be expected. Importantly, if you look at serious treatment emergent adverse events, there was no difference between the two arms. And the reasons for discontinuation, at least by ECRFs, were not different in terms of driving discontinuation of either arm. So with the right prophylaxis and treating the right patients, this toxicity profile is manageable, and we'll continue to explore the kinetics of those toxicities over time as well.